Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome to the Genetics Podcast. I'm really excited to be here today with Dr. Susan Liebman, who is a distinguished university professor emerita at the University of Illinois, a research professor in the Department of Pharmacology at the University of Nevada, and as of November 19th, a published author of an amazing new book called The Dressmaker's Mirror. Susan, you've done yeast genetics research towards the tail end of your career. You got very involved in human genetics, clinical genetics research, inspired by a family tragedy. I would love to start there. If you could talk about your book, why you chose to write it, and give people a little bit of a glimpse into the story, and then we can get into some of the broader themes that you touch on throughout the book of access to genetic testing and screening, and what it means to actually for a family to learn these kind of facts about themselves through some technology that's available to us now.
Well, at the time I wrote the book, it was pandemic, and I was very involved in trying to find the mutation that killed my young niece at age 36 when she just dropped dead. I suspected it was genetic because the autopsy came back and said that she had dilated cardiomyopathy. And my sister, in her 50s, my niece's mother, had been diagnosed with dilated cardiomyopathy. So although the doctors had told us my sister's disease was caused by a virus, and I'm a geneticist, I figured this out. So I was concerned about it and I was working on it. I was involved with finding relatives. It was a focus of my life. At the same time, it was pandemic and I was in a book club and I actually led the discussion of Mrs. Polyfax series books. I don't know if you guys know that book series, but it's a 65-year-old lady who, when I first read it in my 40s, was a really old lady. But now that I read it again in my book club, she was a young lady. Yes. But anyway, she was widowed and unhappy, and she goes to see her doctor and explains that the garden club and the cooking club just don't do it for her. And he says, well, get a life. And she says, oh, yeah, I always wanted to be a CIA spy. And so the rest of the series is her going out spying. But one of the questions that I asked the group is, what haven't you done that you still want to do? And so I had to ask myself that. And I'd had a very lucky life with family, career, and art. What hadn't I done? Well, I always wanted to write a book. I wanted to write a memoir. So I got into writing a memoir about the family, but then I was doing all this other work on genetics about my family. And I realized that I needed to marry those two. And so the result was the book.
It's amazing. It's really well written.
Thank you so much.
I really enjoyed it. You mentioned, your niece who's 36 years old who tragically dropped dead. I wonder if you could tell people a little bit about that so they can get a sense of what that felt like. Like it came out of nowhere, it sounds like. And then the diagnostic odyssey, I guess, and what you unraveled after that so people understand, a little bit more about the broader story of the Weiss family.
So my niece had been married for less than a year and divorced. She met on a place called MySpace. That's a precursor of Facebook.
I remember it.
She met a guy that she knew in high school. And he had also been married for a year and divorced. And he had a crush on her. He joined the women's basketball team just to be near, crew team, one of those teams, just to be near her. So they had this whirlwind romance, and they're 36 years old. They're a little concerned. They want children, but she's kind of old. Will that happen? She gets pregnant. They move the wedding date up. The world is their oyster. They're redoing their apartment in Brooklyn, making a nursery. And they go out to dinner. She's 6 months pregnant to celebrate his 37th birthday. They weren't out to dinner yet. They went out in the afternoon. The dinner was going to be that night. And I have a picture of her that day. She's just beautiful. Blonde hair, blue eyes, just radiant smile with her husband. It's on the cover of the book. They're in this Bogota type restaurant getting little snacks. They ask them, can you fill out this card? How do you rate it? And she says, it's a 10. And then she collapsed. And her husband says to her, Karen, are you kidding? What's happening? And she slides down. He slides down. Well, he knew she was dead already. And she was. They were, within less than 2 minutes, the ambulance was there. It was downtown Brooklyn, but they couldn't restart her heart. They took her to the hospital. They tried to save the baby, but it was only 6-month-old fetus. And they did an emergency cesarean section, but the baby died. And various people saw were there and took Andrew's phone and called family. So family was there at the hospital to meet him and to be there when he got this terrible news. And they called my nephew, who called me. I said to him, Jeffrey, are you kidding? And he says, Aunt Sue, I wouldn't kid about something like this. Karen and the baby are dead.
It's hard to imagine something more heartbreaking than that.
My daughter was also pregnant at the time. She was 3 months pregnant. And so it was terrifying. The autopsy came back, no unusual anything. But New York City coroner's office sent her heart away for further testing. And a week later, the result was what they called idiotropic cardiomyopathy, because there was no reason for it. But there was a reason for it. Right. My sister had cardiomyopathy, dilated cardiomyopathy. And although they had said that hers was caused by a virus, it was clear to me as a geneticist that mother and daughter have these unusual things. It's probably genetic. So, of course, it was terrifying for my daughter as well, because Karen actually did have some symptoms. She had shortness of breath when she would climb stairs, and she had what seemed like a cough. But every 6-month-old lady has this 6-month pregnant, a woman who's 6 months pregnant has this kind of stuff her doctor told her. And you teach 3rd grade, of course you're catching colds. So they weren't at all concerned about this, and they didn't know.
And you describe a moment in the book where, the story changes, it flips, where you go from, the explanation that never really made sense to you of it being a virus to a diagnosis of dilated cardiomyopathy, making the connection to you had other family members, and you start to search and speak to specialists, right? Talk a little bit about that. A lot of people listening to this podcast have been in that situation, either as a rare disease parent or a researcher, where you've got this case where things just don't make sense. And most of the time, unfortunately, people don't get to the bottom of it. So how did you actually get to the bottom of it here?
Well, I guess I was fortunate being a geneticist and being in a genetics community. And so, of course, I talked about this tragedy when I would meet people. How are you? Oh, I'm good. This happened, a lot. And I was part of the yeast genetics club in Chicago. I went there every month. I was well known there. And I was talking with one of my colleagues, Steve Meredith, who said, oh, Steve Krohn's wife is a genetic cardiologist. Why don't you talk to her? So I made an appointment and became her patient. And she took care of me. And what she said was, it's genetic. We don't know what it is, but since it's genetic, we want to check your heart every year with an echocardiogram. And your children and your nephew should also be checked. So she was sort of taking care of the family in a sense as well. But my husband wasn't so happy about this. He was afraid, my children my son wasn't married yet. My daughter was married and was thinking about children, that maybe this would frighten them and we don't know. And so we had a big discussion. And in the end, we agreed. We had a plan. The plan was I could tell them whatever I wanted. The first once I could tell them what they were supposed to do, how to get the echocardiogram and what but I couldn't ask them about it next year.
Right.
Or the year after. or I couldn't nudge them ever again. They were adults. I told them once, then leave them alone. So that's what we did. And the truth of the matter is my daughter followed through and was as crazy, nervous about it as me. Every little twitch she worried about. And my son went to his doctor, and one of the doctors said to him, "Does your mother have cardiomyopathy?" And I didn't. I didn't present with any cardiomyopathy. And so this well-meaning but ill-informed doctor says, "Well, if your mother doesn't have it, you can't have it, so don't get tested anymore." And he never got tested again. But the truth of the matter for our listeners is, you can have the mutation and pass it on to your children and grandchildren. And with the luck of the draw, you never get sick. You don't always get sick because you have the mutation. There's something called penetrance that tells how the chance that you will get sick. So this doctor confused and many do, the fact that I didn't present with disease with assuming that I didn't have the mutation, which it turned out I didn't, but we did not know that. So I was lucky to meet Dr. McNally, and she was not only a cardiologist, but she was a research cardiologist. And she kept testing my sister's DNA, sending it to these fledgling genetics companies. Harvard did it a little, but they only had a few genes, a handful of genes. Yeah. And it was negative. It was negative, negative, negative. Every year it was negative. And finally, she said to me one day, she said, the price of sequencing the human genome used to be $1 billion, but now it's $50,000. And I have 5 patients like your sister who I can't figure out what it is. And I've got this grant. I'm going to sequence her whole DNA, her whole genome. So that's what she did. She sequenced her whole genome, but she didn't tell me the results. So she found when you look at it, you find lots of mutations. You find lots of changes everywhere. How do you know which one? Well, she's a cardiologist. She makes an educated guess. And she found a mutation in a gene that she thought could be the reason. But she wasn't sure enough to tell me. She was sure enough, however, to write a paper. So she wrote a paper with this hypothesis. And then I moved from Chicago. I was no longer her patient. And I kept writing to her genetic counselor, Lisa Castillo, who didn't answer me. In the beginning, she answered, but then I couldn't get any answer. Finally, my sister died. And instead of just writing to Lisa, I also copied Dr. McNally, who wrote back within 10 minutes and told me that she had moved. She and Lisa had both moved from Chicago to Northwestern. And I guess her email was forwarded, but Lisa's wasn't. So yeah, at least email, you just can't trust email. And they told me that they had this paper out that I asked, what happened? We sent you all this blood. And they last heard they were trying to make fibroblasts out of our blood to check out this gene that they thought was the cause. So, of course, I looked up the paper, and when I did the search, I found Frederick Roth, the son of John Roth, who was a yeast geneticist whom I knew, had published a paper finding the exact same gene, the exact same mutation to the letter, very unusual, causing the exact same heart failure.
Yeah.
But the person had checked off, don't contact me. So there was no way to contact that person. But it seemed like this must be a relative. I mean, how did they have the exact same mutation? The other thing we didn't know early on was, maybe I was home free. Maybe it was just a new mutation in my sister. And her child and children had to worry, but not me. But here it was out in the world again.
Right.
And then I kept doing more library research and found a paper by many, many people from all over the world. We're studying this gene, the FLNC gene. And a researcher, Dr. Michael Arad, had 2 patients in Israel who had 2 families, not 2 patients, 2 whole families, who had this also the exact same mutational change. So I wrote to these people, and they wrote back and said, by any chance, are you an Ashkenazi Jew? And I said, yes, because their 2 patients were Ashkenazi Jews.
Right.
And then I looked it up in the IBD, the intestinal bowel
Inflammatory bowel disease?
Inflammatory bowel disease, which was at the time the best and only DNA database. And 1 in 800 Ashkenazis had this mutation and nobody else.
Wow. 1 in 800.
Yeah.
Wow.
But actually, you see, 1 in 200 people have cardiomyopathy.
Yeah.
And half of them have it because of a gene. So if you look in the general population, 1 in 500 of everybody in America has a chance of having a gene that's causing dilated cardiomyopathy.
Yeah.
1 in 800 Ashkenazi have a chance of having the specific mutation in my family.
Yeah.
So I hope I answered some of that without rambling too much.
No, it does. And that, I mean, there's been a remarkable amount of research in the Ashkenazi Jewish community on lots of different, founder event driven genetic mutations. And so this is yet another example of, the profound importance as well of studying populations that had been isolated in the past or are in the present day, because the impact of these otherwise very rare variants in the general population can be incredibly high in this community. And through the book, you talk not just about the discovery of the gene, but also, I guess, your evolving views on genetic screening more broadly. Maybe you could talk a little bit about what, maybe before all of this happened or during all of it, you talked a little bit about, like, it's obviously should be everyone's choice. You need to have an informed decision about what to do. But, I think over the years, you've evolved a view of the growing importance of genetic screening. And it's still, despite the point you made about the Human Genome Project, and today it's a couple $100s, right, to do whole genome sequencing or exome sequencing. But I get the sense we're still far from having universal access, even in, communities where there are cases like this that could, I think the penetrance of this is 15%, right?
Penetrance of sudden death is 15%.
Oh, right.
Penetrance of the is 90%. So
right,
if you go to a cardiologist and they check out your heart, they're going to find something wrong 90% of the time if you have this gene. But you may never notice it, and it's only going to cause you to drop dead 15%. Right. Well, to me, that's pretty bad.
Yeah, it's terrible. What do you think about genetic screening? Where do we need to go as a community from here, based on everything you've learned?
Well, what I've learned is that there's a lot of discussion in the genetic societies, including the American College of General Genomics, also ClinVar, about screening and mutations. It's hard to know if you have a change, because of course we all have different slight changes in our gene, different flavors. It's hard to know if that's going to cause disease or not. But they have a way of categorizing genes. They say it's pathogenic. It's going to cause a problem. It's likely pathogenic. We have no idea. Variant of unknown significance. It's likely benign. It's benign. Okay, so those are the categories. Since you don't really know for sure, doctors get frightened of this. Also, genetics is one of those courses that doctors took in college that they didn't like. It was hard. They can't remember penetrance. They don't want to deal with it. They have a lot of things to deal with. And they remember it cost $1 billion to get the DNA sequence. Well, right. I'm not going to send my patients out to spend $1 billion or even 50,000. They haven't kept up with the lower cost. What I've learned is the American College of General Genomics has many committees, and they meet and decide which genes can we do something about. If we find a mutation in this gene, can we do anything? If we can do something about it, and if they come across this mutation by accident, it's secondary findings, we should tell the patient. So your DNA is being sequenced for cancer, and they find a gene that can cause sudden death that you can prevent. They recommend doctors to tell you about it. But they don't recommend you get screened for those genes yet. They're still debating that, and I don't understand why. The best I can understand is, well, we don't have enough doctors, we don't have enough follow-through, those to me are things we have to change. The other concern that is valid is a scientific concern that if you pick out a mutation in an ethnic group that hasn't shown any disease, but it's a mutation that showed disease in a different ethnic group, it might not have the same effect in that ethnic group. So we have to be aware of those things. But you can now get a test for under $300 that covers all 81 genes. It's not covered by insurance. Not everybody can afford it, but many people can. And I recommend it. And it took some years, but the gene that killed in my family, FLNC, is one of the genes that has been chosen to be in this group. And they fight about it every year. Somebody will write, take it off this card,
Right.
put it on. So it's fluid, it changes. Clinvair has, instead of 81, they have more like 400 or something. So different groups differ in the genes that they've chosen.
If listeners want to go deep into ClinVar and how they make these decisions, I had the pleasure of interviewing Heidi Rehm, who's the chief genomics officer at Mass General, who is the leads ClinVar, for episode 86. So go back in your timeline and look at that. But it is remarkable how fractal this problem is. You find genes, and then within genes, to your point, you find some variants that are clearly pathogenic, others where it's less clear. You have this ancestry, ethnicity challenge that you pointed out. And so I think one of the things the field is struggling with is just the amount of information balloons every year as sequencing becomes more and more ubiquitous and more edge cases pop up. Is it fully penetrant? Is it incompletely penetrant? How do we explain that? And it's a big challenge. And I think it gets in the way, to your point, of doctors knowing how to navigate the system, patients knowing how to navigate the system, because it's hard, it's complex, and it's still not a thing in medical school for the most part, right?
The other thing I'd like to point out is that it's not at all unique. I go for blood pressure tests. They test your blood pressure all the time, every place, whatever you do. If you have high blood pressure, it doesn't mean you're going to die of high blood pressure. It doesn't mean anything, but puts you at more risk. And they put you on some kind of medicine because of that risk. You don't go around spending your life thinking, oh my God, I'm going to die of high blood pressure. The same thing with a mutation. You get an FLNC mutation that can cause sudden death. But now you're getting tested, your heart's being looked at every year. If something goes wrong, they're putting you on a medication. Maybe they'll give you not a pacemaker, something that restarts your heart. Sounds terrible, but dropping dead and dying is a whole lot worse, and it really isn't so bad. Get over the fear of genetic testing telling you something terrible that's going to change your life. It doesn't matter if you don't get tested, you still have the same mutation.
Yeah. It's a very fair perspective. I think it's one of the interesting challenges that the field has of it is treated differently, whether you agree or not, than almost every other medical measurement. I think some would argue that there are things that you can find out through these tests that you can't do anything about, and people may make an informed decision that they would rather not know and live with that.
Right. And they're not part of these 81 genes.
Yeah.
They're not part. Yeah. And I fully respect that. I'm not even dealing with those kinds of things.
Yeah.
I'm not encouraging that. The only thing I'd say about that is if you had something terrible like Huntington in your family and there's nothing you can do about it right now. But if you wanted to have children and marry, you can do pre-implantation genetics and be sure that you don't pass the gene on to a child. So there are possible reasons for wanting to know these kinds of things.
Yeah, I think that's absolutely right. We have this discussion about actionability, and you can define that very narrowly as clinical actionability, right? Can you do something for me? But I think often the aperture of actionability needs to be widened. Can you do something for the next generation? Can you do something that's not medical, like live your life differently? If I were pre-genetically counseled and said, look, there's a chance you may find out you have the Huntington's gene. In my personal circumstance, I would probably like to know, because I'd live my life very differently if I knew I had it, even if there was nothing to do about it. Somebody else may make that decision totally differently. But I think there's different spheres of actionability that we can think about that aren't just medical. But I very much understand the need to do no harm as well from the doctor's perspective.
Right. Well,
you can't control all these things.
I do describe the story of the very famous family, Wexler, the daughters saw their mother die of this terrible disease, and they couldn't face the knowledge that they would have it. But instead, one of the daughters went out and found the first disease gene, Nancy Wexler, created an army of scientists who mapped the gene. And her sister, Alice Wexler, wrote a book. It's 25 years old now. I used to use that book in my genetics class to inspire students, called Mapping Fate. And that also inspired my book, because it was the story of a family who suffered a genetic disease. You understood how it affected the family, and they also did something about it. It didn't turn out to be something. Neither daughter decided to take that test, but it was available for the rest of the world.
And it was the Huntington's gene that
It was the
Nancy
Huntington
Wexler
gene.
had
That was the
discovered,
first
right?
gene ever mapped.
Wow.
And yeast genetics was used to help map it. So what we learned in yeast genetics was the epiphany of an idea that David Botstein had of a way you could map genes, and they used it.
The first research project I was ever involved with as an undergraduate was working to help build a mathematical model of the mitotic spindle in yeast in a lab at University of North Carolina. And there was a wet lab, dry lab. I eventually found my way into human genetics. I spent about 3 months in yeast cell biology, but you spent most of your career in it. I'd love to talk about that.
Right.
What did you work
on? I worked with a man named Fred Sherman, who, as a graduate student, and I don't know if you know Charles Yanofsky in E. coli. Well, Charles Yanofsky helped create the relationship between a protein and a gene. He showed that if you change the DNA, this is before DNA sequencing, so they had all these tricks, you would change the amino acid in the protein. And Fred Sherman, instead of doing that in a bacteria, he proved that the same genetic code was working in a eukaryote, that is, an organism with a true nucleus. That turned out to be very similar to human cells, even though it's this little baker's yeast, single-celled organism. So I worked with him, and then I worked on translation and the genetic code and stop codons. And it turned out that there was this mysterious thing called a psi factor. It didn't seem to be inherited 2 to 2, the way normal good genes were. It was inherited by all the babies, 4 to 0. And some years later, Reed Wickner helped to turn out that this was a prion. And what I can tell you about a prion, the first prion was the mad cow disease prion that Stan Prusner tried to promote. And there was a lot of opposition. It's a virus. The way a prion works is you've got a protein, and its normal shape is like this, and it's all over the place, and it's all good. And then it changes shape. And when it changes the shape, it aggregates. So it attracts other molecules of that protein. And every time there's a new one born, it attracts it. And so it's not out here able to do its job. It's stuck in this aggregate. And so our work on yeast prions helped prove the prion hypothesis. So that was very exciting.
Fascinating.
We showed that a chaperone protein, there are proteins that help other proteins that aggregate, disaggregate, was essential for the prion in yeast. And that helped prove that Prusner's idea of a prion in humans was correct. And then it turns out that other diseases, Alzheimer's, ALS, have aggregating proteins, and there's some connection between the aggregation and the disease. And so we've been putting human disease genes for ALS, for A beta, into the yeast and studying the factors that affect its aggregation. And it's toxic in yeast. And mutations that rescue toxicity in yeast, the same homologous mutation rescues toxicity in humans. So we're able to learn a number of things in yeast. Nowadays, with the availability of CRISPR and all sorts of things that you can do in humans, younger scientists have a lot of times switched into using human cells.
There's a researcher that you may, are you familiar with Sonia Valab at the Broad Institute?
No, I know the Broad Institute.
So she, when she was younger, her mother died in her, I think, early 50s from a kind of mysterious dementia. Turned out to be prion disease.
I do know her.
Yeah. She's one of my dream guests for the podcast. She has a very, very, in some ways, kind of parallel story to yours. So she discovered she carried the mutation. She has dedicated her life as a researcher to working on this disease in humans, and they've done some amazing work both in kind of
And her
therapeutics,
husband was
genetics, but
along working with her. They chose
that's
that
right.
area to study.
Exactly. Yeah. So for everyone listening as well, go Google them and check it out because it's incredibly amazing research and inspiring story that's landed in a lot of places. Yeah, it's reminded me of that. I didn't realize that was the area of yeast biology that you worked on, but it's interesting. It's very
By accident.
translational.
By
Yeah.
accident. I didn't choose this wonderful area. It chose me, and I was very lucky for that to happen. But
Yeah.
that's the way science sometimes works. You just
Absolutely.
follow happy accidents.
How it usually works. Going one more step backwards in time, you were an undergraduate at MIT, and I think while you were there, it was basically right during a transition period where in prior years women were in the university but were not actually on campus in dormitories, and you were one of the first classes to actually live on campus. You were still a huge minority, I think, on the campus, 50 women to 800 plus men. I'd love to hear a little bit about like your decision to actually go there, knowing that you were going to be greatly outnumbered, but also just a little bit about how the institution was at the time and how it's changed because it's obviously was and has been for a long time one of the best institutions in the world for math and science. But like all the other institutions, it's changed a lot over time, especially in how it treats women scientists and engineers.
My parents encouraged me to be the best that I could and to reach whatever potential I could. But they really thought that I should be a teacher. That would be a terrific thing to be a grade school teacher and to have a family. My sister went to college in the Boston area. She was at Tufts, and she discovered that there were women at MIT.
Yeah.
My sister was boy crazy. She was, her whole life was this different dates and whatever. And I was very shy and studying all the time. And she said, MIT, that would be a good place for my shy sister. She'd meet boys. I mean, there would be only boys. She should apply there.
Well, that's a great reframe, actually. You're not outnumbered. You've selected a cool. Yeah, continue. That's an interesting reframe.
At that time, there were no computers and stuff, and we had these poor secretaries trying to write out your transcripts. And so, and copiers didn't exist. So you were allowed to apply to 3 colleges from my high school, Erasmus Hall High School, which is a huge high school, plus the Brooklyn College, plus the city schools you could apply to. And so I chose MIT as my. It was supposed to be a reach school, a regular, school that you should get into, and a school below what you thought you could get into. And so MIT was my reach school. And when we went to interview, that seemed like a real school to me, whereas the other places looked like party places. I didn't get it. So I really was excited about it. And then my senior year, I had great grades, but there were 2,000 students in my class. So it was very competitive. But what pulled me ahead was my entering a science fair. At that time, it was called the Westinghouse Science Fair. Now I think Intel runs it. And it's the Cadillac of all science fairs. And I didn't win. I was semifinalist. I was one of 40 finalists who were sent to Washington, D.C. And there was publicity and TV and letters, and people invited me to join scholarships to their schools, and MIT admitted. So that was an exciting thing. And I still have connections from those people that I met in those years. We had an amazing high school teacher who encouraged us to apply to the Westinghouse Science Talent Search. He taught, but he called AP Zoology, but it was really a front class. If you applied, you didn't have to take the final.
Right. And if you Google your name and Westinghouse Science Fair, it's there, on the internet, believe it or not.
Yeah.
What did you do? I
have a picture on the cover of my book of me standing on the steps in Washington, D.C. There's some Nobel Prize winners in that group.
Wow. What was your project?
My project was the similarities and differences of Micrococcus lycodicus and Sarcina flava. So these are 2 bacteria. I took a course at Cornell for this in the summer, and we learned how to do lots of different bacterial tests on different bacteria. And one of the things that the professor mentioned was that these two bacteria seemed to be misclassified. They were very similar, but they were classified as different species. So I asked him to send me these strains, and I decided to use every test I learned in the lab that summer, if I could do them at home, to see if they both had it or not. That was basically the project. You could not win a Westinghouse and Intel Science nowadays with a project like that in your kitchen, but those days it was good enough. And yeah, when I made the agar plates, they all got moldy. So my dad said, well, why don't you go to there's a lab supply house. So I went down the street to the lab supply house and I asked to buy it. And the owner said to me, why? And I said, I'm doing this Westinghouse Science project. Oh, you're a girl. You don't even know what you're doing. Don't waste your time. That's for geniuses. You can't possibly do this. So I went home crying, and my dad was furious. He said, what kind of idiot is that? And he found a place that sent me these plates. And then when I was a sophomore in college, he sent me a newspaper article where this guy was arrested for falsifying scientific data at his place. So there, he was a jerk.
Yeah, he was certified jerk. Wow.
Yeah.
So you said your parents wanted you to be a teacher, thought you'd be a good teacher. How did you end up? But you clearly were great at science from a young age. How did you decide going to MIT? You thought you wanted to be a scientist, and then, or were you not really sure and just following?
No, I was going to be a teacher. I went to MIT to be a teacher. I forgot to look that MIT didn't offer any education classes. And my parents forgot to look at that. When I came home one summer, they tried to have me take education classes, but it never worked out. And in the end, what I realized was what I loved about teaching was finding new information. And I did become a teacher. I became a researcher and a professor. I just, when I was in first grade, I wanted to teach first grade. When I was in sixth grade, I wanted to teach sixth grade. And so it went.
Yeah.
So.
Yeah, and how many students, postdocs have you helped teach or mentor over the years? Probably quite a few, right?
Yeah, maybe up to 25.
Yeah,
and of course, I've worked with undergraduate students in classes where I had 125 each time I taught the class.
Yeah.
Yeah.
As we wrap up here, do you have any advice for early career scientists? You've had an amazing career. When you look back, what advice do you have to people who may be it can be early, middle, or late in their career. It doesn't matter. Wherever you feel like you think you have something to share.
I would say follow your loves. If you love something, follow it. I remember at some point people told me they were going to quit science and quit biology and go into computer science because that was where all the money was. You don't know what the future is going to bring, but if you follow your heart and do something you love, whether it turns out to be fashionable and you make a lot of money or not, you still at least have done something you love. And you can change if it doesn't work. You can nowadays we figured out people can change careers and change direction. It doesn't have to be for life, but do something you like. And if in writing for grants, that's one of the big downs in my career. You have to write for a grant. If you don't write, you're definitely not going to get it.
Yeah.
If you send out 5 grants and you get rejected in 4, it doesn't matter. You got the 5th. And you have to ignore the failures and focus on the successes.
Yeah, I like that. There's this great article. I don't know who this comes from, but it's called The 4 Types of Luck. And, when people think about luck, they tend to think about a dice roll, right? And that's type 1 of luck, which is, can you roll a 6 or not? But there's other types. And you're referring to type 2, which is luck from motion. If you put more applications out there, if you do more stuff, you're going to get more results, right? So sometimes just you can talk yourself out of submitting the application because nobody ever wins these things. But then, if you submit 10 of them and 10% of people win these things, then you create your own luck in that way. I think it's a concept that's I've always found really interesting because it's true, right? There's blind luck is
true, but
small part of it.
Certain personalities, every time they get a rejection, it brings them down. It brings us all down, but you have to ignore that. That doesn't count. You throw that out. That's a nothing. What only counts is the one that says yes.
Yeah, I love that. Well, thank you. This is an amazing conversation.
Patrick.
I will put a link to where people can order your book in the show notes. By the time this will be out, it'll be available anywhere you like to buy your
I
book.
just want to say one last thing. I'm sorry
Yes.
about my family.
No, please.
They have some great things about life. So my dad used to tell me when I was always worrying in high school about, it was so competitive, more competitive in high school than at MIT for me. He would say, do your best and leave the rest.
Yeah.
And every night, do your best and leave the rest. We love you just the same, whether you get an A or an F. And my sister, as a first grade teacher, used to meet these parents who wanted their kids to be great soccer players or great whatever. And
or the president. Yeah.
Yeah. And that wasn't going to be what they were. And she would say to them, celebrate your child. You have a wonderful child. Find what's good in your child. Celebrate your child.
I love that.
And other such things that stick with me from my family are in the book that inspire me, and I hope will inspire or help others. So.
Yeah, and when we were talking before the show, you shared something that I thought was really a great way to think about it, which is that even when you have you inherit a mutation that could be life limiting or life changing, you can live a happy and a great life, right? And you don't have to necessarily let that get in the way. Maybe you could talk a little more about that.
Well, I think there's this little proverb about it's not how many breaths you take, but the moments that take your breath away that determine a wonderful life. And we have to, and my dad, actually, his father died when he was 40. And of course, he lived through the Depression and the war, and he had a lot of troubles. That's why to him, the other thing he would say to me was, just learn to be satisfied. If there wasn't an earthquake today, you should be thrilled. Just be satisfied.
Yes.
So that was his other view. But when he was a student teacher, he had two jobs. He both taught in high school and he opened his own business. But when he taught in high school, one of the colleagues was 50 years old and died suddenly of a heart attack. And everybody was in shock, of course. And he, having had his 40-year-old father die not too long before, said, well, at least he lived a good life, a long life.
Yeah.
Everybody loved my father, but they were so mad at him. He was not used to having people angry with him and think he was callous. He thought about that the rest of his life. And when he turned 50, at a family celebration of Passover Seder, he stood up and said, he hanged the glass and he said, well, now I can say it. I'm 50. My bags are packed. I'm ready anytime. I've had a great career. I have a wonderful wife. I have children. I'd love to see them grow more and have more grandchildren, but I'm ready. It's good enough. And he lived another 16 years after that. But every year he would repeat that.
I love
and
that.
I think he believed it.
Yeah. It's a great note to end on. Buy the book, read the book, enjoy it. Susan, thank you. This was so much fun and inspiring, and I learned a lot from you. So thank you.
Thanks so much, Patrick.
That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah who produced the show, and James Pierce from Selected Frequencies who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.